So, today we will be talking about selenium or Se, and the role of selenium in human health, sort of the investigations that were related to it were accelerated during the 1980s, and now we have a better picture about the metabolic activities of selenium. Not only that, but also how it relates to the fact that whether our diet is rich in selenium or not, to some extent depends on where crops are grown, in the sense of what the soil is like. So, in general, this is sort of true for some of the minerals, but also for selenium. And if you live in a geographical area where the soil is not rich in selenium, then there is a tendency to have lower intakes of selenium, and this then influences health in a very strong way. And this has been shown to be the case in China, and we'll talk about a couple of diseases that are associated with low selenium intakes in general. So, just getting started with this, selenium is a mineral, as you know, and in general, selenium is found bound to amino acids, which of course make up proteins in general. So, if you were to go back and look at the structure of an amino acid in general, you can depict it as a carbon in the middle, okay, and a hydrogen on one side, and COOH on one end, NH2 on the other end, and here is the side chain or the R chain. So, that is a general structure of an amino acid. But the amino acids that selenium is actually associated with are cysteine and methionine. So, if you were to look at the structure of cysteine in general, so here is the structure of cysteine with the C here, and then if you want to, an NH2 here, COOH here, and an H there, and a CH2 here. And then typically speaking, cysteine would be an SH right there. However, if selenium is replaced here, then it becomes SEH, and this is called as selanocysteine. And because of the fact that the group, the selanocysteine, the selano group is actually available, so this is considered to be active form of selenium with regard to metabolism. So, this is selanocysteine. There is also what is referred to as selanomethionine, that is, selenium can be associated and become a part of the structure of the methionine, which is also an amino acid. So, if you want to look at the structure of methionine, and this becomes COOH, and there is an H there, and this is like CH2, and here it is SEH and then CH3. However, in this case, this is selanomethionine, and because of the fact that selenium is bound between two carbon atoms, this selenium is not as actively available in vivo, and so comparing the two of them, selanocysteine is the more available form. And then ultimately we also have what is called as selanophosphate, and that is a structure that can look like this. So, and this form actually then is the one that is responsible for presenting selenium so that it can be incorporated into amino acids and then into proteins. So, the remarkably different thing about selenium is that it's incorporated into amino acids, becomes a part of the amino acid first, and then becomes a part of the protein. Then this should then make you realize that the incorporation of selenium into these amino acids then has to happen at some point with regard to the translation of proteins. So, keep in mind proteins are made, there's a process for them to be made, and the first step in making a protein is to be able to copy the DNA sequence into an mRNA sequence, which is the transcription part of it, and then the mRNA leaves the nucleus and goes into the cytoplasm, sits on the ribosomes of endoplasmic reticulum, and that is where the protein or the peptide chain is made, and tRNA is a molecule within the cytoplasm that actually carries the correct amino acid onto that growing polypeptide chain. So, keep in mind tRNA in general are specific to amino acids, and they bring specific amino acids into the ribosome so that that amino acid matches with the sequence in the mRNA and is linked into the growing polypeptide chain. What are good sources of selenium in general? Overall, organ meats, seafood, muscle meat, grains and cereals are good sources of selenium. As I mentioned before, the amount of selenium in plant-based foods depends on the soil content of selenium in general. There is, in terms of the bioavailability of selenium, there is not really a consensus in terms of which forms might be more bioavailable in general. However, we do know if we are consuming selanomethionine or selanocysteine, and this would mean that automatically you're talking about the food that contains protein in it for us to consume these forms of selenium, then they're supposed to be absorbed almost completely, because what happens is in the intestinal tract during the digestion of proteins, you end up creating amino acids, di-and-tripeptides, and usually they're taken up, absorbed pretty well. And selenium in its inorganic form is also present, and it's called selenite and selenate. And selenide. And these two are the inorganic forms more likely to be found in plant-based foods, and their absorption is somewhere around 50%. So, we absorb relatively more selenium from the animal-based products in general. And then once after absorption takes place, as you know, the next step would be from the enterocyte, you absorb these nutrients, and then they go into the capillary bed vessel that exists inside the lamina propria. From there, finally, the capillaries merge into and make what's called as the portal vein. So, what is actually absorbed sees the portal vein and then the liver first. But in general, selenium is transported in the form of selenium proteins, which are shown up, or rather selanoproteins is what they are called, and you can see with the acronym there, it's referred to as SEPP1. So, transfer proteins for selenium would be SEPP1 and also glutathione peroxidase. Glutathione peroxidase is an enzyme that converts peroxides into harmless water. So peroxides can promote the risk of oxidative stress in general. So glutathione peroxidase is an enzyme that converts that harmful peroxide into the harmless water. So these have very important functions in terms of maintaining oxidative health, within the cells in general, but it also acts as a carrier of selenium along with the selanoprotein 1. That's how selenium is transported in the circulation. There you go. So your textbook has information about how selenium is incorporated into the proteins with regard to details about what the structure is with regard to tRNA, and how there is the coming together of various proteins in the making of that, in the incorporation of selanocysteine and selanomethionine into proteins. You don't need to know all of the details in it, except for the fact to keep in mind that the active form of selenium, which is selanocysteine, has to be incorporated into proteins, and that's where its function lies. Okay? So then moving on, what happens to the metabolism of selenium? And you have a lot of detail in there, but this is what I wanted to focus on in general. So if you were to look at this as on this side of the part, you have the cell, and here it is the plasma. And so you're exactly what is going on is that, as we mentioned before, you have selanoproteins. So this is extracellular selanoproteins. And then you also have selanomethionine, because keep in mind this is not as much of the active form, so it survives in that form within the circulation. But once selanomethionine then enters a cell, it can be converted into selenide first, and then into selanophosphate. Okay. This reaction is catalyzed by selanophosphate synthetase, which converts selenide to selanophosphate. Now keep in mind, from the extracellular proteins that contain selenium in them, as they enter the cell, keep in mind, through proteolysis of that protein, you can end up making cysteine. So during proteolysis, this is released just like any other amino acid can be released, and so it's available to the cell. However, a lot of the selanocysteine doesn't stay, so this is just the free selanocysteine, meaning just the amino acid by itself. But in general, this does not stay free for too long, because of the fact there is an enzyme by the name of a lyase which converts this to this very quickly and efficiently. Once you have selanophosphate, then what can possibly happen is that this, along with the tRNA molecule that contains selenium. So remember how I said tRNA molecules are specific to certain amino acids, and they work as transporters of that amino acid, come and join the ribosome and help add that amino acid to the growing polypeptide chain. So, selenophosphate and this together then end up making selenoproteins. And then these selenoproteins endogenously also can be hydrolyzed to make free selenocysteine in general. So, essentially overall what you need to get from here is that the concept of how selenium is put into proteins as well as the other metabolic products that are helpful in making selenium available to the cell as there is a need. Now, as I have already said, then what are these proteins that have selenocysteine in them, and how do they work? So, for example, one of the enzyme systems that contain selenium in them is glutathione peroxidases, and that is what shows up right here. It is not just glutathione peroxidases, but also fatty acid, I am sorry, it is not just glutathione peroxides, I am sorry, the enzyme is glutathione peroxidase, and they convert peroxides to harmless water. So, these peroxides can just be regular peroxides, or they can even be fatty acid peroxides, because keep in mind fatty acid oxidation is something that happens biologically, but too much of the oxidation or unregulated oxidation of fatty acids can lead to the formation of peroxides, and that can kind of then induce oxidative stress over a period of time. So, we have the enzyme systems called glutathione peroxidase, which converts even the fatty acid peroxides. The peroxide part of it is converted into water. And then there is another group of enzymes called thioredoxin reductases. Thioredoxin reductases. So, what is the concept? The concept here is the word thio, Thio, and what thio or thiol, either way, what this means is this is referring to the sulfhydryl groups that are present in a lot of proteins in general. And what you need to know about this is that proteins that have sulfhydryl groups play a very important role in reducing oxidative stress, without any doubt at all, because they have the capability of donating this proton or the hydrogen and reduce the formation of free radicals in general. So, this class of enzyme thioredoxin reductases have the ability to kind of so they provide they have the ability to provide the hydrogen group to a lot of enzymes that do various things in the body. So, essentially, for example, enzymes that function in DNA synthesis as well as cell signaling use these thioredoxin reductases, which help reduce by their enzyme activity, they reduce certain parts of those proteins, because keep in mind enzymes are proteins, and that is how they keep them functional. Another important selenium containing protein is obviously selenoprotein, and the main reason, the main function of this is to be able to transport selenium throughout the body, and also to supply selenium to the brain and to maintain normal neurological function. When there is selenium deficiency, then selenoprotein levels go down, and it can be used as an assessment marker for the status of selenium in general. And then finally, iodothyronine deiodinases. So what is, there is a class of enzymes, they are deiodinases, meaning they remove iodine from certain proteins, and the protein that contains iodine that we know of, or the hormone, is thyroxine. And so keep in mind, we have two forms of thyroxine, we have T4 and T3. T4 has 4 iodines in it and T3 only has 3, but the conversion, but T3 is the active form of thyroxine. So, the conversion of T4 to the active form of T3 requires an enzyme that has selenium as a structural part of it. Requirements and recommended dietary allowances. I am going to let you take a look at this. So, in terms of selenium deficiency, as I have said before, a couple of deficiency disorders have been identified in China. The first one is referred to as Keshan's disease, and this, in general, there are two types of outcomes that you see. If there is an acute, if selenium availability is acutely low, then there is insufficient heart function. But if selenium availability is low over a prolonged period of time, then this leads to heart enlargement and cardiac insufficiency, as a matter of fact. But the selenium supplementation in these people was helpful, but it did not correct the insufficiency of the heart. And as a matter of fact, the important finding, or the research based finding that has to do with this disease, is the concept that there is a Coxsackie virus that actually causes this cardiac insufficiency. However, they had found that if you take this, so let me go back for just a second. So, as I had mentioned before, there is what is known as Keshan's disease that is seen in China, because the soil there is poor in the amount of the selenium. And if selenium deficiency is very acute, then you end up having what's called as insufficient heart function. However, if the selenium deficiency is prolonged and it happens over a period of time, then there is heart enlargement and varying degrees of cardiac insufficiency. They also found that there is a Coxsackie virus that promotes this this phenotype of this cardiac insufficiency. And what researchers found was that if you were to take the Coxsackie virus, if you isolated it from patients who had the Keshan's disease and introduced that into mice, then the Coxsackie virus became much more virulent, that is much more stronger. So, in other words, in people with iodine deficiency, then the Coxsackie virus becomes more virulent or causes a much more stronger form of the disease. And so, if the patients are then, not the patients, in general, if people are iodine sufficient, then even if they get this Coxsackie virus, they are not likely to get as sick, is the point. And then the other condition that happens because of selenium deficiency is called as Cauchin-Beck disease. And essentially this is sort of like an osteoarthritis. It's a chronic disease, And because of the role of selenium in developing healthy chondrocytes, and chondrocytes are cells that give rise to cartilage in general. So, you can realize that if there is selenium deficiency and if you are not producing the proper amount of cartilage or even healthy cartilage, then which exists essentially at the tips of the long bones to sort of keep the friction to a minimum, then as a result of that, if you don't have enough selenium and not making enough healthy cartilage, then you may end up having a problem with arthritis type of problems, as you can say. However, in these patients, if you provide them selenium, it doesn't work as a treatment, and now so researchers suspect there are things other than selenium that actually kind of promote this phenotype in general. Okay.